Welcome to this video introduction to flow cytometry. In this video, we will explore what flow cytometry is, how it works, and its application in measuring cell surface molecules. We will also discuss some characteristics of flow cytometry. Let's get started. Do you often come across the following brightly colored images and wonder what they are about? The pictures below are actually obtained by flow cytometry. Flow cytometry, the abbreviation of which is FCM, is literally understood as flow, fluid, cyto, cell, metry, measurement. So flow cytometry refers to a technique that detects a single flowing cell, thus responding to the physical and chemical characteristics of the cells, such as the size, granularity of the cells, and expression of antigenic molecules. The great invention of flow cytometry combines several wisdoms of disciplines in a clever blend of computers, electrical engineering, optics, fluid dynamics, mathematics, organic chemistry, and other subjects. So its invention is also not originated from one brilliant idea of a sole scientist, but from many individual discoveries, which came together to create the current flow cytometer. Mac Fulwiler was an American biophysicist who invented the first flow cytometer in 1965. The instrument allowed for the rapid and accurate analysis of individual cells. Robert Auer and Louis Kamensky, on the other hand, were two American biophysicists who together invented microfluorimetry in 1959, a technique based on the principles of fluorescent labeling and microscopic observation, which allows for the rapid and accurate analysis of individual cells. In addition to the above, many other scientists have also made contributions to the invention of flow cytometry. So how is a flow cytometer composed? Although there are different brands of flow cytometers on the market, such as BD, Beckman Coulter, Thermo Fisher Scientific, Biorad, Miltenyi Biotech, etc., their main components are no more than three components: the fluidic system, the optic system, and the signal detection and analysis system. The fluidic system is the key that allows us to detect cells one by one. We make a single-cell suspension of the cells to be tested and stain it with a specific fluorescent dye labeled antibody. The single-cell suspension enters the flow chamber under the pressure of a constant gas. The buffer without cells is ejected from the sheath tube under high pressure, and the direction of the inlet of the sheath tube forms an angle with the sample flow to be tested. The sheath fluid flows around the sample flow at a high speed, similar to the principle of laminar flow. The cells to be tested are arranged in a single row under the envelope of sheath fluid and pass through the detection area in turn. The optical system of a flow cytometer consists of two components, the excitation optics and the receiving optics. The excitation optics, laser and lens set, focus and adjust the laser light so that it always intersects the cell at the same angle in the laser detection zone of the flow chamber. The light emitted from the cell is divided into three groups, one for forward scatter, FSC, one for side scatter, SSC, and one for fluorescence. The forward scattered light is received by the receiving lens and then converted into a current signal by a photodiode and recorded by the electronic system. The emitted side scattered light and fluorescence are received by the lens set and then pass through a series of filters to be received by specific photomultiplier tubes, or PMTs. These filters allow only a certain wavelength of light to pass through, while matching the peak emission of a specific fluorescent dye. Filters that allow a specific spectral band to pass through are called band pass filters. Earlier we mentioned forward and side scatter. The degree of light deflection depends on the physical properties of the particle, especially on its internal complexity and its size. Cell morphology, surface morphology, membrane, nucleus, and granularity all affect light scattering. Forward scatter light is the measurement of diffracted light in the direction of the laser light path or advancement, proportional to the cell surface size or cell size. Side scatter light is the refracted and reflected light collected from an angle almost perpendicular to the laser light path and is proportional to the cell size or internal complexity. A good example of this is shown below, where the x-axis direction represents FSC, with the smaller cells represented by points appearing further to the left of the axis. The SSC, or y-axis, is used to distinguish between cells of similar size but different cell size, with the more complex cells appearing further to the up of the axis. Finally, let's talk about fluorescence, which is produced by laser excitation of cells with fluorescent dyes. The most used laser is the argon ion laser, which is capable of emitting light at a wavelength of 488 nanometers and can excite a variety of fluorescent dyes. The two most widely used fluorescent dyes in combination are fluorescent isothiocyanate, or FITC, and phycoerythrin, or PE. The absorption spectra of FITC and PE peak around 495 nanometers and can be excited by the 488 nanometer wavelength of the argon ion laser at the same time. When monoclonal antibodies are combined with fluorescent dyes such as FITC and PE, then they can be used to identify specific cell types that express specific surface proteins. Thus, in a sample with a mixture of multiple cell types, different fluorescent dyes can be used to distinguish among different cell subpopulations. Combined with data from FSC and SSC, the fluorescence expression of each subpopulation can be used to determine the type and number of cell subpopulations present in the sample. This also explains what is expressed in the pictures at the beginning of our video. When the cell and laser intersect to produce SSC light or fluorescence, an electronic signal or voltage pulse signal is generated. The generated scattered light or excited fluorescence then enters a photodetector, PMT or photodiode, and is converted into electrons. The electron signal is superimposed to form a current. The current signal is amplified and converted into a voltage pulse signal. The voltage pulse signal increases as the cell intersects the laser, peaks when the cell enters the center of the light, and then gradually returns to a base value until the cell passes through the light. Finally, the voltage pulse signal is quantified by the signal processor into pulse height, amplitude, and area. This numerical data is then transferred and stored to the cytometer's computer workstation for further analysis. Flow cytometers are extremely fast and can analyze millions of cells in seconds and can measure multiple parameters from one single cell population at the same time, such as cell differentiation, counting of various types of cells, and analysis of cell phenotypes, with the advantages of high speed, high precision, and good accuracy. But the power often means that the instrument itself is expensive, and a laboratory may need to spend several $1,000,000 to purchase a flow cytometer, which requires specialized personnel to operate the instrument at the same time. In addition, we usually do not need so many functions, which leads to the waste of resources. In conclusion, flow cytometry is the result of the wisdom of many scientists, and the power of flow cytometry has made it widely used in various fields of biological and medical research, such as immunology, developmental biology, cell kinetics, physiology, cell biology, molecular biology, etc., with significant applications. Thank you for watching!